The 1918 influenza pandemic affected almost 30% of the global population and killed over 50 million people. There are still many unanswered questions about the 1918 flu pandemic, but investigations on the history of this pandemic have provided a framework for early detection of contemporary flu pandemics. Let's consider some of the properties that distinguished the 1918 flu pandemic from seasonal flu outbreaks and why they can alert us of flu pandemics today. What was unusual about influenza in 1918? I don't need to remind you that flu occurs every year. This is typically just one flu season a year that in North America peaks in the winter months of December, January, and February, and then tapers off by the end of March. One of the first clues that something was amiss with influenza in 1918-1919 was the timing of the flu outbreak. The 1918 flu had 3 peaks of flu-related mortality in a 12-month period, in early summer, fall, and late winter. A harbinger of a flu pandemic is a change in the seasonal pattern of disease. In fact, it was a change in the predictable pattern of flu-like illness that alerted public health officials to the 2009 H1N1 flu pandemic. A second unusual feature of the 1918 flu outbreak was the number of infected persons who died. Influenza certainly causes its share of deaths on an annual basis, but the death rate during the 1918 flu was over 10-fold higher than expected for flu. Influenza viruses typically replicate in the upper respiratory tract and cause the signs everyone is familiar with: a runny nose, a sore throat, sometimes a cough, generalized fatigue, and muscle ache. The 1918 influenza virus caused pneumonia, a disease of the lungs. Differences in clinical disease can be due to changes in the genetic makeup of the virus, which allow it to infect a different type of cell. The incidence of very serious disease and the high mortality rates observed during the 1918 flu pandemic suggested that something had changed in the influenza virus itself. So was the influenza virus that caused the 1918 flu pandemic different? We determine how similar viruses are by comparing their genomes. Influenza viruses encode the genes that they need to reproduce on individual short pieces of RNA. There is tremendous diversity of influenza viruses in wild bird populations. A few strains of influenza virus are also resident in pigs and humans. Occasionally, an influenza virus from a bird infects a pig or a human. The avian flu virus can exchange whole genes, a process called reassortment, with influenza viruses already present in the pig or human. This provides a ready-made way to produce a virus with a completely different set of proteins, which is a much faster way for a virus to evolve than relying on the slower process of accumulating mutations in a gene. Our ability to resist infection with influenza virus requires that our immune systems recognize specific sequences in virus proteins. If these virus proteins change, the virus can escape immune detection. Influenza viruses do evolve by mutation, which is one of the reasons you need a flu shot every year. When a whole new protein appears because the flu virus has acquired the gene that encodes it from another flu virus, remember this is called reassortment, vaccines and our immune defenses don't work. The 1918 flu virus was apparently new in the human population, although there are no convincing data on its animal origin. This means that the global human population was subjected to an influenza virus for which there was no immunological defense. Everyone was susceptible. And the virus also had properties that enabled a more severe disease than the seasonal flu. The lessons learned from the 1918 flu pandemic are that a change in the seasonal occurrence of flu is an indicator of a potential pandemic. This is why public health officials pay close attention to reports of flu cases each year. Additionally, it is important to sample the influenza viruses in animal populations to look for evidence of changes in virus genetic makeup, which could allow a new flu virus to infect humans. Surveillance of annual flu cases and virus genetics in animals needs to be conducted globally, and all information must be shared because influenza pandemics are global health concerns.